Hello, everyone. Welcome to session 3. We are excited to hear talks and poster highlights on immunotherapy and emerging treatments in kidney cancer. My name is Katie Beckerman. I'm at Vanderbilt University, and I would like to introduce my co-moderator, Eric Kaufman from Roswell Park. Before we get started, briefly, I'm going to send out some trivia questions to get everybody excited. I'm going to go through these somewhat quickly, see if I can.
Katie, this is Eric. While you're getting those ready, just like to say I'm honored to be joining yourself and moderating this session over the next hour and a half. Just to give people an idea what the format's going to be, we're going to start off with 3 talks from some esteemed researchers we're lucky to have joining us for this first hour. And we're going to hold questions from ourselves or the audience until the end of all 3 talks, after which we hope to have about 15 minutes for questions before we start part 2 of the session at 3 p.m.
And here's our last trivia question.
You're not doing it. You're not presenting next. I'm going to be back.
I don't know if that last one came out, so I apologize, but maybe we'll go ahead and get started.
Okay, without further ado, I'd like to introduce our first speaker, Dr. Charles Drake, who is presenting research out of Columbia University Medical Center on combination immunotherapy without anti-PD1.
All right. Well, thank you for that introduction. I'm delighted to be here. Hopefully everyone can see my slides. Maybe somebody could say yes.
No, we don't see your slides.
Yes, I do.
All right. So it looks like the slides are up. So today, these are my disclosures. I used to consult for many companies, actually, but recently I accepted a position in leading immuno-oncology at Janssen, actually, and so all these things will go away. What I'd like to talk about today is the broad topic of modulating the kidney cancer tumor microenvironment by blocking the innate cytokine IL1beta. I'm going to start out by talking a little bit about how this works in cardiovascular disease and some hints that this might have anti-tumor activity. Then I'm going to talk about some preclinical work combining this with anti-PD1 in kidney cancer and a little bit about a clinical trial that's testing that. Then I'll talk about really interesting data, and these are preclinical data that are recently accepted for publication in clinical cancer research, and this is about combining immunotherapy that's not PD1. And the immunotherapy that's not PD1 is anti-IL1beta. And I'll show you some data that's really neat. It really combines very nicely with the TKI cabozantinib. Could be even synergistic. And then at the end, I'm going to tell you a little bit of a story about the cells in the kidney cancer TME and tell you that they're much more complicated than we think, actually, both in mice and in patients. So this is like kind of what's eventually going to be an old school view of the tumor microenvironment, and even now it's vastly simplified. The idea is that within the tumor microenvironment in the kidney cancer, or in other cancers, there's multiple cells, and these are the suppressive cells. On your far left, and my pointer doesn't work, so you have to just use your eyes and listen to my words. So if you look on the bottom left, right, there's regulatory T cells. These are CD4 T cells that inhibit anti-tumor immune responses from CD8 cells. One pathway many pathways are involved in Tregs, but one of particular interest involves adenosine and the A2A receptor. If we go up to the top right corner, there you see a population of cells called granulocytic MDSCs. These are related to neutrophils. They are very difficult to distinguish from neutrophils. They probably are immature neutrophils that don't die. Immature neutrophils have a short half-life, actually, but not these cells. These cells are attracted into tumors through interleukin-8 and a CXCR2 pathway, and these are particularly suppressive cells. If you go down one slot on your right, you'll see monocytic MDSCs. These are, as their name says, related to monocytes. They express CD14. They are probably attracted by IL1beta, and IL1beta is kind of a broad spectrum cytokine in that it maintains the presence of many of these cells. Finally, on the bottom right, there's a population of cells called tumor-associated macrophages, M2. This is even more of an oversimplification. These cells, you can distinguish them in mice pretty easily. In humans, these are suppressive cells that are related to macrophages. They express CD163 and other markers. They are real, but it's not a single thing. It's sort of a spectrum. And I'll show you in later slides in the talk that this presentation, or this sort of presentation of what's in the TME, is almost certainly oversimplified. So this is how we got interested in IL-1 beta, actually. So there was data, and there are data in cardiovascular disease, that the inflammation mediated by innate cytokines like IL-1 beta causes heart attacks, quite simply. And so there was a clinical trial of a drug. It's called Canakinumab. And this is a trial to see if the drug stops patients from having a second myocardial infarction. Cardiovascular trials dwarf ours. So this was a trial of about 10,000 patients using placebo and then 3 different doses of canakinumab. And you know, you could read this paper. It's a nice paper, but bottom line, it works. That is, blocking IL-1 beta prevents second heart attacks. But in this trial, there was a big worry because immunotherapy in the immune system prevents cancer, right? And so there was a worry that if you blocked IL-1 beta, like probably happens when you block TNF, you might see an increase in cancer. And so the trial included a tabulation of cancer, although it wasn't specifically designed to address those data. But nevertheless, when the trial was completed and published, it was shown that the incidence of cancer was dramatically decreased in a dose-dependent fashion in the patients treated with IL-1 beta blockade. So if you look at the middle bar, that's the hazard ratio for all cancers, you can see that as the dose increases, the hazard ratio for cancers decreases, so that at the highest dose of IL-1 beta blockade, there's about half the relative risk of cancer. And given that there are thousands and thousands of patients, this is, of course, statistically significant. It's even more dramatic for lung cancer. So if you go to the bottom of the slide and you look at the relative risk of lung cancer, at the highest dose, the relative risk of lung cancer was 0.23, again, statistically significant, suggesting that IL-1 beta might have an anti-cancer effect. And this is supported by some other studies as well. So we sought to model this in kidney cancer using what is, I admit, and the reviewers of this paper would certainly agree with me, is not the best model for kidney cancer we have. There are other ones being developed by multiple groups, including Billy Kim and James Brugarolis and several others. But we didn't have those yet. So we basically used Renka for this model. And you can see a figure in the center and the bottom that you've seen many times before, right? So this is a study of growing tumors. They're in black. You add IL-1 beta blockade, it's a little better. You add PD-1, it's a little better, and you do both, and it's a lot better. And this is showing the changes in the tumor weights. And again, this is not, of course, unique to IL-1 beta blockade. You can show this with LAG-3, TIM-3, many, many other things. What's different, though, is kind of how it works. And so if you look at this next slide, we did carefully, and this is, by the way, this work is all done by a really excellent fellow. His name is David Agate. Unfortunately, he left Columbia and went to join Memorial Sloan Kettering across town because they were shinier. And what Dave and other folks in the lab showed is that this kind of therapy doesn't really affect the T cells in the tumor all that much. So there wasn't a big change in the gross number of CD8 T cells, in the number of CD4 cells, or even in the regulatory cells. But this was a bit of a myeloid therapy. So IL-1 beta decreased those granulocytic, those PMN MDSCs. It really didn't affect the monocytic MDSCs too much. But what it really did, and this is probably how we think it works, is it increased the number or the percentage of good macrophages, the M1s. And the best way to show that is by looking at the ratio of good guys to bad guys. That is the M1 to M2 ratio. And you can see that in untreated tumors, it's about 1-to-1, maybe 2-to-1. But when you combine IL-1 beta blockade with PD-1 blockade, it's dramatically increased. What's next? What's next is the thing that should be next, frankly. And we were fortunate to receive support from Novartis for this. Dave wrote this trial, and now Matt Dallos, a faculty member at Columbia, is running this trial. So we have an ongoing neoadjuvant trial combining anti-PD-1 with anti-IL-1 beta in patients. This is a trial where patients get three doses, and then they receive a radical nephrectomy actually it's 2 doses, I misspoke, 2 weeks after that second dose. And we have great data, and I won't show it to it because it's not done yet, but the bottom line is that we're doing both flow cytometry and single-cell RNA sequencing to see if what happened in the mice happens in the patients. That is, if you see an increase in M1 to M2 ratio, actually. And I can tell you, we do, actually. In the first couple of patients, we've seen that, but I'm going to leave that to Dr. Dallos probably to present in the future. What we also did is we modeled something else, and I think this is the cooler part of the paper, frankly, the more interesting part. So if you look at the NCN Guidelines, and I'm sure that you don't have to look at them because you're kidney cancer experts, you probably know this all by heart, but the first line regimens typically include either a TKI anti-PD1 combo with pembrolizumab plus axitinib, nivolumab plus cabozantinib, soon to come, blah, 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 TKI monotherapy, and then also the possibility of combination immunotherapy with anti-CTLA-4, ipilimumab plus nivolumab actually, which is a reasonable combination. But the majority of the patients currently probably aren't getting cabozantinib in the first line, and they probably are getting an anti-PD1. So what happens in the second line? Many of these patients are eligible for cabozantinib, which is a good drug. Response rate is solid. It's in the 20 to 25%. It certainly makes sense. So what we wanted to model is not giving more PD1, is to model whether cabozantinib, which does have clear myeloid effects, added to or synergized with IL-1 beta blockade. These data are a little bit better, actually. So if you look on the bottom left at the tumor outgrowth curve, once again, you see IL-1 beta having about the same effect as it did before. And then you see an effect of cabo. But check it out on the bottom in that yellowish color. There you see a really nice combination effect, potentially beyond additive. We didn't do the math, so I don't want to say it's synergistic, but it definitely looks different than either agent alone. And if you look at the tumor weights on the right in colors, again, you can't see my pointer, but both IL-1 beta blockade and cabo have an effect in these tumors, as they should. And the combination has a more pronounced effect, which is good, right? Once again, this is really mostly not a T-cell kind of therapy. So if you look in the top in the bar graphs, none of the treatments appear to dramatically affect the bulk CD4 and CD8 cells. I should mention that anti-PD1 does increase the effector CD8 T cells, and cabo doesn't really make that worse. That's not shown here. They do. Both cabo and cabo with IL-1 beta does decrease the regulatory T cells a little bit, actually, so that's true. Once again, you see that this is likely a myeloid effect. So if you look on the bottom, IL-1 beta decreases the granulocytic MDSCs. Cabo doesn't really help. It's about the same. But what's really I hope you remember the last graph I showed you. So look on the far right on the bottom. We had to alter the scale because the ratio of M1 good guys to M2 bad guy macrophages here is in the 20 range. That's fairly high, actually, and that really shows that this combination effect can repolarize at least, hopefully, the macrophage part of the tumor microenvironment, actually. And again, this effect is more pronounced than it was with PD1 plus IL-1 beta. It really looks like the cabozantinib, the TKI, is adding, or potentially, again, even synergistic with IL-1 beta blockade in this setting. We also did a lot of work trying to figure out what happens on the cytokine level. And I'll just point out a few things. It gets hard without a pointer. But one thing is we wondered was if there's not big differences in the CD8 T cell populations, are they changing their cytokine secretion? Interferon gamma is predominantly a T cell cytokine. And we thought maybe, like, in that combination, you're going to see more interferon gamma. And if you look on the top left graphs, you see actually that's completely not true. We didn't see increased interferon gamma. We saw a decrease in IL-1 beta. That's good because it's you're blocking it, right? We saw a little bit of a decrease in IL-6. That's on your bottom left, the next graph over. But what's really neat, actually, and I hope this is true in patients, is that there are recently papers published by Malero and by others showing that IL-8, interleukin 8, is a prognostic or predictive factor for PD-1 therapy in patients, right? Casey Grow actually is one of several murine analogs of IL-8, and it looks like maybe this is downstream of IL-1 beta and cabozantinib. So it could be that you can fix multiple problems in the TME by blocking IL-1 beta in combination with cabozantinib. And that's why I highlighted that. And there's other IL-8 analogs you can see in the paper, actually, that were downregulated as well, suggesting, honestly, which is neat, is maybe IL-1 beta is like an upstream kind of thing. Here's where it gets complicated, and look, this is just the way it is. I apologize for reality, okay? So I showed you this really neat picture of like, what are the, what's in the TME, and I can tell you that that's not really true. So this is an experiment where we used 28 color flow cytometry to stain the TME for both myeloid and T cell markers. And what we did is we did this in one big panel, and then we deconvoluted it with clustering, unbiased clustering, to see what kind of cells are actually in there. So this is like what the whole, on the top left is what the whole TME looks like. And then you can see what the different treatment groups look like. And you can kind of already get an idea that they do different things. So different treatments affect different cell populations. If you look on the left side on the bottom, these are all the myeloid cells. So the F480 cells are shown to the left in like orange. Those are the top blob. And within that top blob in the next figure, there are clear subpopulations and a lot of them, okay? So the F480 population is very diverse. The Ly6G, you can see in the far left, is the orange blob at the bottom. That's actually made up of 3 separate clear populations: the brown one, the magenta one, and the red one, which are just distinct populations. If you go over and look at the T cells, so move over to the right, look at the CD4 T cells. That's the middle kidney-shaped blob, ironically. If you look to the right, that's made up of very clear subpopulations of CD4 cells. The CD8 cells are the bottom blob that looks like a shoe. That's also made up of 3 distinct subpopulations. This is not sequencing. This is actually at the protein level by flow. And the bottom line of this is that there are multiple very clear subpopulations of both T cells and myeloid cells in the TME. How do these associate with treatment? So these are the clear populations, actually. So these are the granulocytic populations on the top. There's really only one major monocytic population, 2 DC populations, and many actually macrophage populations. And what's neat, but also kind of daunting, is that only a couple of them are associated with treatment. Most profoundly, this population number 20 looks like it's the good guys. It's the M1 macrophages, which go up and turn red with Cabo and Cabo plus IL1beta. So you can see that at the bottom. I'm not trying to daunt you with this complexity, but I think that's really what the truth is. And I'll show you that it's the truth in human samples. So these are ongoing studies led by Alex Obradovic in the lab, a wonderful MD PhD student. And what we've been doing is we've been analyzing the TME of kidney cancer patients, focusing on both the tumor and the adjacent normal, and in addition, carefully sorting out the CD45 positive versus negative cells. So we've been getting 4 populations per every patient, and then we've been analyzing them with single-cell RNA sequencing them. And we've also been doing some fairly advanced systems biology that Alex, who's a systems biology MD PhD student, is leading. And this is just to show you that what I showed you in the mice is true in people. And that is that there's more populations than you probably were picking ahead of time than you were thinking of. So there's multiple NK, natural killer populations, multiple B cell and T cell populations. But what's interesting, and this is probably too small to see, if you look on the bottom right, only a fraction of them are overrepresented in the tumor versus the adjacent tumor, and only a fraction of them are associated with stage, actually. We did find one population of macrophages that is associated with stage. And also, if you look in the next slide, we developed a signature for that. And if you apply that to a small set of samples, you can see that that macrophage signature beautifully correlates with recurrence. So on the bottom, you have patients who have not recurred, and the majority of them have this signature. And then on the bottom row, you have patients with no recurrence, and most of them do not have that signature. So this signature is strongly associated with recurrence. I should point out that this is an incredibly small sample size. It's what we can get from Columbia, 8 patients. But recently, our friend Brian Reaney and Scott Hack from Vanderbilt got us a much larger data set to which we've been applying this signature, and preliminary results suggest that this is a beautiful prognostic signature. So I think I'm going to try to finish on time, which is unusual, which is good. What I showed you is that blocking IL1beta reprograms the myeloid component of the tumor microenvironment in kidney cancer patients, and we'll see if it does the same thing in patients. I showed you that it combines with anti-PD1. You should not be particularly surprised by that. What is more interesting to me, I think, is the notion that combining a non-PD1 therapy with cabozantinib might be interesting. And IL1 beta is not the only thing to think about. There's anti-IL-8, which is a drug that's under development by another group of folks at BMS. There are adenosine might reprogram that, adenosine A2A blockade. There's multiple other ways you could think about doing that. But instead of doing it with anti-PD1, right, maybe the combinations to think of involve the TKI cabozantinib and potentially other TKIs. I showed you an oversimplified view of the cells in the TME, and then I told you that it's not really true. Frankly, the cells are really markedly diverse, and I think we really need to step back and think about our new profiling tools, both single-cell RNA sequencing and also flow-based. Finally, the people who did the work, I tried to say their names during it. Dave Agin did the majority of the animal modeling. It's a beautiful paper, which is finally accepted in CCR and has a lot more data than I had time to show. Alex helped with the systems biology for that, and he's leading the human single-cell RNA sequencing work. The master of the flow is Casey Ager. He's a postdoc in the group, and the clinical trial leader is Matt Dallas. Finally, I would say that we're really lucky to have many, many great collaborators, both at Columbia and previously at Hopkins. Most recently, our friend Brian. I mean, I think Brian and Scott helped us out with a series of RNA sequencing samples that will allow us to really see if our macrophage signature validates. With that, I will finish, and I think we have questions at the end. Thank you for your attention.
Thank you so much for that great talk, Dr. Drake. I'm going to transition to the next talk. Please continue to send in your question and answers, and we'll hold those for the end. But I wanted to introduce our next speaker is Dr. Timothy Chan from Cleveland Clinic, presenting a talk entitled Genetic Mechanisms Underlying Immunotherapy Efficacy.
So thanks very much, and it's good actually that Chuck ended on the note of some molecular determinants. I'm going to go ahead and continue that discussion here and under the section for biomarkers for immunotherapy. And what I'm going to talk about really will focus on genetic mechanisms underlying immunotherapy efficacy with a emphasis on some of the leading areas of signal for renal cell carcinoma I think Chuck did a great job in beginning to show us sort of some of the more novel out-of-the-box molecular determinants that are probably at play in renal. This is definitely a different factor, different disease than the usual, and I'm going to try to put that into context along with some of the other major topics going on in immunotherapy biomarker space. So these are my disclosures. I have not gone into industry yet, so these are still applying. Now, the tumor microenvironment, as Chuck had begun to discuss, is quite complex. What is really happening is this is a dynamic rheostat of a number of different types of cells, including what you show here, tumor cells, along with a bunch of different types of immune cells, an entire ecosystem here, which really works together to try to eliminate or deal with this foreign issue, foreign lesion, as you will. And a number of things actually, a number of major biomarkers are in the space. And many of you are pros here, so you'll have known this already. But for those of you that aren't, there are a number of biomarkers that are either approved or almost approved or in the process. And I sort of list them here, and by no means is this comprehensive. But we have sort of this neoantigen mutation space here that basically is a reflection of non-self. And as we know, the immune system is geared towards defending against non-self, which cancer is. And also, PD-L1 and PD-1 expression, obviously, as you know, this is approved also in certain cases. Not as helpful as it could be in renal in different kidney cancers, but all the same, there's a pan-cancer signal for this. And within this category, I also include things like different signatures and sort of this concept of a hot tumor as well, which definitely is in play, and also other potential resistance drivers and mutations for immunogenicity. And of course, there's a lot of different other things that can interact and determine which way the scale falls, either pro-rejection or anti-rejection, microenvironmental alterations in innate immune system, microenvironment, and so forth. So it can get pretty complex. Our opinion has been in the lab that much of this falls under the purview of genetics. We try to boil down the question and try to answer the question that, what you see from mutations all the way to anti-tumor immunity taking place at the cellular level is actually the result of a genetic program. And when you have successful anti-tumor, elimination by the immune system, it is because there are certain, thresholds, certain things being satisfied. And that is, not only does a tumor cell have to be recognized as foreign, either through some type of foreign antigen, neoantigens, or self-antigen, what have you, or ERVs and so forth. There's a lot of things in play here. But the right things have to be presented. And furthermore, the T cells have to have the right phenotype, non-exhausted, right, which is, as you know, partly what anti-PD1, anti-CTLA4 does. And there has to be successful ligation here in this trimeric complex of MHC cognate, peptide, as well as TCR that recognizes it. So what does this mean in terms of biomarker space? We have done a lot of work, as you know, previously on, this here. You can't see my actual pointer here, but here on the tumor cell space, trying to identify what exactly is being seen as foreign. There's been a lot of work, as you know now, on TMB, not the perfect biomarker, but certainly something that we see as potentially a metric or a foundational finding showing that mutations can be seen as foreign by the immune system and can underlie the treatment effects, successful treatment effects of immune checkpoint blockade. I'll just present a little bit of data in terms of how broadly this can be applicable. We did at Memorial sequence almost 2,000 patients with either ICI or almost 9,000 patients, slightly over 9,000 patients who were never treated with ICI, all advanced tumors. This is pan-cancer treatment when you look at it together, and you can clearly see a difference here on the left-sided panel in terms of mutation burden. When you have a high mutation burden here, defined as over 17 or so mutations, you can actually see a clear difference. And I'm not showing you this data, but you can clearly see a dose effect response. But when you actually try to break this data down, right, you break this data down, most of the tumors that are high TMB really fall within this concept. And exceptions are a glioma. And also, I don't show here, renal cell carcinoma doesn't really work for renal. And since this data, other prospective trials have actually shown this to be the case. Now, there's other determinants that are potentially important that can really, sort of make TMB go really high, right? So such as MSI, very rare, of course, but once in a while you do see this, or some other DNA damage repair mutations. Recently, we've had some HRD defects as well that can result in a high level of indels. And there, what you can really see, as I show here by the original publication by Luis Diaz, is that if you have MSI patients in pan-cancer settings, you can still get a nice response. And hence, that's why MSI and also, high TMB is also used, potentially applicable in RCC, albeit at the frequency is very low. And just to update everybody, genomics-based anti-PD1 approvals are a reality now. This is a list of what has been approved so far. MSI positivity, both in colon cancer and pan-cancer for hypermutated, tumors, as well as TMB high, are approved right now. But in general, what I would say is that TMB is not that effective of a biomarker for renal cell carcinoma. So the question then is, what is? And I would like to get into a discussion now of other aspects that we think may be important. All right. So if you see here, I'll go back up to the previous slide. You see here, if you look in the green box, the orange thing really is tumor mutation burden. Okay. But you see that The blue squiggle is actually HLA, and HLA, as we know, is one of the most diverse genes in the human genome. Yours differs than mine, likely differs than people in different parts of the country. It really is something that evolution has driven in human beings to be very, very diverse. And so a little bit about HLA diversity. There's a long and sort of glorious history of looking at transplant antigens. It started, previously with many folks ending up with Doherty and Zinkernagel winning the Nobel Prize. What they had identified is HLA as being a determinant for transplant specificity, but also they posited, but they were unable to show at the time because of lack of technology, that specific HLA genes, the more resistance you have, the more diversity you have, the more resistance to susceptibility to pathogens like HIV, malaria, COVID, right? Perhaps, may occur, and this is termed HLA heterozygous advantage. And so what you can actually see here, and so we tried to answer this question to see if this is what you see in cancer, because if in cancer you also have foreign elements, if it really is a foreign lesion defined by novel foreign epitopes, the same type of biology should be in play, right? So you see here a picture of MHC plus neoantigen complex. We measure TMB using that little red arrow up there for cognate epitope, binding as a ligand, but the receptor itself is the MHC molecule. We asked the question, okay, well, if other people like Mary Carantine and others have seen that heterozygosity or maximal heterozygosity just at the allelic level for HLA can really boost survival of patients with HIV from getting AIDS, well, can we see this in cancer? And the answer is yes. We put together data sets of over 1,000 patients and above showing that zygosity can matter. But that there is context specificity. In renal cell carcinoma, as I will show you before, it tends to have a bigger effect, especially in low TMB tumors. And then the second panel on the right, you see that if you have a high TMB and high mutation and high HLA diversity, you can really start separating the differences. So we were able to show this in a Nature Medicine paper last year, that they are independent predictors. Here, these are all melanoma patients, and if you take high TMB and high what we call HED or evolutionary diversity, it's a molecular metric that we've measured. You can actually show that patients do better if you have both, which is in panel C. And in fact, this is not dependent on just heterozygosity. If you have fully heterozygous patients, remember all different alleles on every single seat in MHC class 1 DNA space, what you can really see is that this still matters, right? It's still dependent. And why is this? Is because the higher your HLA diversity along the x-axis here, the more peptides you can bind, right? So it's really all in the math, right? On the one hand, with TMB, you have a lot of big antigen space. The universe of peptides that are available that you can bind to present is bigger. But on the other hand, you have to have a high receptor diversity to be able to present those. And so really, we're putting together these sort of more novel ideas now for, of course, a lot of things happen downstream, right? A lot of things happen downstream in terms of, activating T cells and so forth. But if there is no immunogenic rejection potential, all right, if the potential is not there, the recognition of foreignness is not there, it ain't going to happen, right? So, and we think that this is measured in part and determined by TMB, as you see above, as well as how evolutionarily healthy your immune system is as determined by your HLA diversity. So moving on, some of this space, Chuck mentioned this as well. Combination immunotherapies are all the rage in renal cell carcinoma right now. You see some of these really great trials by Moser, Brian Reaney. Many of you have been involved in these, Monty Powell, Tony Chouari, and others have run these excellent trials showing the efficacy of combination immunotherapy. We really see a strong signal. This work really is transformative, right? So when I started, as many of you did, in oncology patients, renal cell carcinoma was like a rock, right? And finally, we're seeing response rates that we can take home. In most of these trials, again, just like in our own analysis, TMB, and again, RCC is a low TMB tumor, right? And accordingly, TMB doesn't really drive a separation. There have been very nice work like Chuck showed, but also Dave McDermott in his trial, basically showing that there are signatures, including angiogenic signatures and myeloid signatures, that really track with survival. We asked, whether some of this is determined by genetics. And so Bob Motzer, Joe Lee, Ari Hakimi, and I, we helped run this trial and analyze this trial in collaboration with Merck and SI. Now, this is what you see here, the results, biomarker results of the RCC component. This is not yet published, submitted, that HED is probably the major genetic determinant in levo-pembro treated RCC patients. So you see here, very excellent response rate, right? So in the rainfall plot, you see about a 76% ORR, official response rate, 96% overall tumor shrinkage rate. We looked at TMB, didn't matter. We looked at signatures, didn't matter. What really mattered in our, at least this cohort, was the fact that your receptor diversity is higher. So what you do is you can break this by the mean. It says not even optimized, by the mean here with progression-free survival. Higher HED, which is the blue curve, separates out the better responders. But most importantly, it's durability of response. And if you see the bottom plot here on the right and the bottom, there is a dose dependence. It's just like your immunopeptide effect. The more your diversity, the higher you track up with 2-year progression-free survival. So what could potentially be going on at the cellular level? I thought Chuck really did a great job in dissecting that. In our case here with Ipi-Nivo, and Ari has presented this data a few weeks ago at another meeting. What we have really found was very interesting. We found that it's not simply successful patients with CRs, which we've single-celled analyzed after being treated with Ipi-Nivo. It's really related to expansion of this tissue-resident T-cell population, which is really interesting and unexpected, actually. That's in the middle, this really little cloud of purple here. And what we see using pseudotime analysis, basically, is that these resident T-cells actually, when patients do well, they take on this NK-like phenotype, right? This teal phenotype, instead of going down the exhausted phenotype. And we think that that's linked to a good response. In fact, if you tabulate all of the different types of changes in untreated patients, Nivo exposed but non-responsive patients, and responsive patients, you see that it's the dominant signal tracking with response. And you can actually break down some of these signatures and relate them to different angiogenesis and adenosine signatures and myeloid-differentiation. We see much of the same signatures that Chuck has seen already, but this may actually be related to uniquely to this tissue-resident population. So with that, I'm going to go ahead and end. Acknowledgements, of course, mainly for my MSK laboratory. Really sort of amazing people. Many thanks to Ari, of course. And Bob Motzer, Jolie, and others working on renal with us, and also my new lab at Cleveland Clinic and Collaborators, of course. Thank you very much. And we'll wait for the end for questions.
Excellent talk. Thank you, Dr. Chan. As our final speaker for part 1 of this session, I would like to introduce Dr. Vladimir Kolenko from Fox Chase Cancer Center, who's speaking on genome-wide loss-of-function screening for TKI resistance mechanisms in clear cell RCC.
There was probably miscommunication because initially my talk was announced like PARP inhibitors. And indeed, we did some work with PARP inhibitors in collaboration with Alexei Tulin, who is currently at University of North Dakota. But we published an overview of our studies in urological oncology recently. So if someone is interested in our work on PARP inhibitors, it's available through PubMed. Today, I will talk about our experience with CRISPR-Cas9-based genome-wide loss-of-function screening, which we use to identify cellular factors contributing to TKI resistance in clear cell renal cell carcinoma. So, as you all know, traditional chemotherapy and radiation therapy, they are largely ineffective in the treatment of all RCC subtypes. Therefore, introduction of tyrosine kinase inhibitors was like a kind of revolution in the treatment of kidney cancer. You all know about that, but I would like to emphasize role of TKIs just because every time I submit a grant application, at least one reviewer, maybe two, will say, who cares about TKIs now? You have to study immune checkpoint inhibitors. This is the future. But still, TKIs are widely used as first and second line agents for advanced RCC, and for most patients with metastatic RCC, treatment with several lines of TKIs remains the standard of care. Moreover, TKIs can be used in combination, of course, with immune checkpoint inhibitors, and they also can be used as alternative regimen for patients who cannot tolerate checkpoint inhibitors. But despite therapeutic progress, complete response with TKIs have been noted only in a few cases, and initially about like 20% of patients, they don't respond to TKIs. And therefore, to address this unmet medical need, we used CRISPR-Cas9 loss-of-function screening in the hope to identify genes which are involved in resistance to tyrosine kinase inhibitors. So I'm amazed by this technology because we all use CRISPR-Cas9 probably at certain points. But here we used a whole library which was targeting 18,000 different genes, 18, with 5 sgRNAs per gene, so totally like 90 Ks, in one cell population. So initially we infected cells with Cas9, selected cells, selected clones with highest expression of Cas9. Next, we infected cells with a library targeting, as I said, like 18,000 different genes at low multiplicity of infections. So the idea was that some cells will be not infected at all, but other cells will get just one copy of one sgRNA. Next, we again selected this population and induced Cas9 by doxycycline. And later, we cultured cells for about 12 days for 5 to 6 passages, either in medium alone or in the presence of sunitinib. And the idea was that actually cells with knockouts of genes involved in sunitinib resistance will be eliminated from the population. Therefore, sgRNAs will be underrepresented. And using immunosequencing, we will be able to detect underrepresented sgRNAs, and this will tell us which genes and gene products are critical for Sutent resistance. So our research identified a number of genes which potentially can be involved in resistance to sunitinib, and we specifically focused on genes which were not known to be involved in resistance to tyrosine kinase inhibitors. And we found pharyngeal transferase and pharyngeal transferase targets among top hits. Pharyngeal transferase is an enzyme which adds pharyngeal group to certain proteins. It consists of 2 different subunits, alpha and beta. It's a heterodimer. And to validate our findings, we induced knockdowns of pharyngeal transferase B subunit in 2 renal cell carcinoma cell lines. Both are clear cell renal cell carcinoma cell lines. One is 7860, which is long-term cultured, established cell line. Another one was PNX cell line, which was established here at Fox Chase from a clinical specimen, which represented aggressive form of clear cell renal cell carcinoma. And on your right-hand side, as you can see, this is a result of tunnel assay apoptosis. Sunitinib itself, we used sunitinib at concentrations which were described for tumors, for CCRCC tumors, and concentration of sunitinib is much higher in tumors compared with plasma. And as you can see, sunitinib itself induced about 10% of apoptotic cell death in both cell lines. But when we treated cells with knockdowns of pharyngeal transferase, we got much higher numbers for apoptotic cells. For 786 cells, it was about 65%, and for PNX cells, it was more than 80%. So the next step was to see whether we can find any pharmacologically or clinically relevant inhibitors to inhibit pharyngeal transferase. And we searched several databases, including clinical trials. And we found that there are 2 drugs which are currently under clinical trials, tipifarnib and lonafarnib, which are pharyngeal transferase inhibitors, which mainly were used to treat progeria, but also they are used in combination with other chemotherapeutic agents for the treatment of solid and hematological malignancies. And as you can see, treatment with lonafarnib potentiated anti-tumor response of sunitinib in several different CCRCC cell lines. We use like 3 cell lines, HRC 45, 78, and 51, to show that the effect is not cell line specific. And moreover, on the bottom panel, you can see on the left-hand side that we also tested lonafarnib in combination with pazopanib. And in both cases, when we use sunitinib and pazopanib, lonafarnib potentiated anti-tumor activity of these 2 TKIs against like 2 cell lines, PNX and 786. And on your right-hand side, at the bottom, you can see results of tunnel assay, apoptotic assay, again, like a combination of lonafarnib and sunitinib at pharmacologically relevant concentrations, I would say, could induce high level of apoptosis in CCRCC cells. And we examined like synergistic activity or between like lonafarnib and TKIs. We used TKIs which have been approved for the treatment of kidney cancer, sunitinib, sorafenib, and axitinib. And we used, there is program, cells were treated with escalating concentrations of either TKIs in the presence or absence of lonafarnib. And we used CompuSyn software to calculate level of synergism. As you can see, like numbers between 0.4 and 0.6 represent synergism, and whatever is below 0.4 represent a strong synergism. And indeed, combination therapy with lonafarnib and all these TKIs showed a strong synergy against 2 RCC cell lines, 76 and PNX. So next, we decided to see what is the mechanism of potentiation of anti-tumor activity of sunitinib and other TKIs in the presence of lonafarnib. Lonafarnib, pharyngeal transferase actually, participates in lysosomal biogenesis. And by inhibiting pharyngeal transferase, we can suppress lysosomal biogenesis in tumor cells. And lysosomal sequestration of different agents is one of well-described drug-resistant mechanisms. It happens because drugs which are present in free base form, they can easily pass lysosomal membrane, but due to acidic environment in lysosomes, these drugs, these molecules become protonated, and they're trapped inside lysosomes. Therefore, cells can concentrate up to like 1,000-fold higher concentration of drugs in lysosomes compared to cytosol. And lysosomal sequestration, actually, of various anti-cancer agents prevent their accessibility to intracellular target sites. And moreover, lysosomal sequestration induces cross-resistance to various anti-tumor agents, including doxorubicin, methoxytron, and several TKIs like sunitinib, sorafenib, and pazopanib. So to see whether indeed like inhibition of pharyngeal transferase can interfere with lysosomal sequestration of sunitinib, we used immunofluorescence staining, and sunitinib is fluorescent compound. Therefore, its cellular uptake and intercellular distribution can be easily monitored. So as you can see on your left-hand side, sunitinib was accumulated mainly in acidic vehicles, which was obvious by combined staining, merged staining with lysotracer. But at the bottom, you can see that when we used combination of sunitinib with lonafarnib, we got more diffuse cytosol staining compared with cells treated with sunitinib alone. And in the middle, indeed, you can see a graph which shows a number of vehicles per cell in cells treated with sunitinib only or cells treated with sunitinib in the presence of lonafarnib. So we assume that actually lonafarnib prevented lysosomal sequestration by inhibiting lysosomal biogenesis, although like total sunitinib level was unchanged on the right-hand side. These are results of flow cytometry studies, because again, like sunitinib is fluorescent compound, so we could easily monitor its uptake. And as you can see, like lonafarnib didn't affect intercellular accumulation of sunitinib, yet it changed its cellular distribution. And in light of our encouraging in vitro studies, we decided to do, of course, like in vivo experiments. We established PNX xenograft tumors in SCID mice and treated cells with sunitinib, lonafarnib, or combination. As you can see, combined treatment with sunitinib and lonafarnib basically completely reduced growth of xenograft tumors in SCID mice. So, as a summary, what we can say that first of all, we validated the method itself, and we used it to identify molecular factors involved in TKI resistance, and we identified pharyngeal transferase among top hits. And treatment with inhibitors of pharyngeal transferase, we also used tipifarnib in some of our experiments, which worked as well as lonafarnib. So these inhibitors, they prevented lysosomal sequestration of sunitinib and other molecules, and increased anti-tumor activity of TKIs in both like cell and animal models of RCC. And our studies indeed revealed a strong synergistic effect of combined treatment with lonafarnib and several clinically relevant TKIs, including sunitinib, sorafenib, and axitinib. So what we plan to do next? First of all, we would like to use the same approach to see if we can identify molecular factors involved in resistance to other targeted therapeutics in kidney cancer and prostate cancer. In kidney cancer, of course, it will be like temsirolimus. And we also we would like to see if we can identify cellular factors which inhibition would be synthetically lethal with mutations which are prevalent in kidney cancer, like VHL, PBRM1, BEP1, and SETD2. So we can specifically target clear cell renal cell carcinoma and spare normal cells. So special thanks to my collaborators, Peter Machov, who actually was a research assistant professor in my lab and who did most of this work, my clinical colleagues, Rob Uzo and Alex Kutikov, Christoph Seeger and Jason, they helped me to develop the assay, and they shared a library, CRISPR-Cas9 library, and Ilya Serebrisky, he helped us with analysis of our data. And the results of our studies have been recently published in British Journal of Cancer. Thank you.
Thank you so much for that very interesting talk. And now we're going to open it up to questions. Just as a reminder, there's a question and answer box in the bottom left-hand side of your screen. So if you haven't had a chance to send in your questions, we would welcome that. And I'm going to start with a question for Dr. Drake. You clearly in your preclinical models showed some really nice responses in the myeloid populations and both your combination looking at PD1 with IL1B as well as the cabo with IL1B. And a question that we have is, you see a shift in the M2 to M1 in both of these drug combinations, and your multispectral flow can kind of clearly see that differences in the myeloid population with the PD1 and IL1B, but maybe a little bit less so, or maybe it's a different mechanism with the cabo and IL1B. Would you maybe describe a little bit more if you think it's because of the complexity of the myeloid immune suppressive microenvironment? And would you ever foresee putting all these together as a triplet combination?
It's a great question, actually. So you know, obviously we couldn't show all the data, but what was a little surprising to us is we know that you know, there's this family of kinases, tyro, axel, and MER, that are involved in the myeloid components of the tumor microenvironment, especially in kidney. And when we started these experiments, we thought that the effects of cabo would overlap with those of IL1 but the you know, both the flow and there's RNA sequencing that we did as well showed that that wasn't true, actually. And so I think that that was a little bit of a surprise to us. I also kind of, as usual, want to talk that short. I oversimplified the T cell piece. There are changes in the T cell components of the TME when you look more deeply, actually true. And then the other part of the question, which was a very good one, actually, was, you know, would you foresee a trial? And I can tell you that, yes. We've been trying to convince, well, when I was still at Columbia, we were trying to convince our partners at Novartis to go forward with a combination trial. the big question is in the second line, right? What do you do with the anti-PD1? And one idea is that, like, look, it failed already, so why keep it on board? But another idea was like, well, it's maybe doing something actually, and you don't want to lose that pressure of CD8 T cells, so maybe keep it on. And so the trial that we were trying to do is the combination of those three things: PD1 blockade plus IL1 blockade with Canakinumab plus Cabo versus Cabo alone. What's holding it back, besides COVID, which is holding freaking everything back, is that Novartis actually did believe the data that the lung cancer data. And they have a series of 3 phase 3 trials called Canopy that are underway in lung cancer. And I think that, you know, if those trials read out as positive, that will definitely propel enthusiasm for additional trials with IL-1 beta blockade in other cancers like kidney cancer, which we hope they will.
Chuck Nizar here. Great talk. Just want to tell you that Novartis is moving forward with a trial combining Cabo and anti-IL1 beta in RCC in the second line.
Huh. Well, they must have dissed Columbia. We sent them an LOI a long time ago, but that's good.
Dr. Chan, I might direct a question at your work, which was also very exciting, trying to understand biomarkers and immunotherapy efficacy. One of the questions that's come up is you show that very nice single cell data that looks like that the CR population of patients sit right next to the resistant patient population, and that all of these are maybe involve tissue resident memory T cells. I wonder if you could discuss a little bit more about what is differentiating those two populations, and were you surprised that they were so closely related?
Yeah, that's a great question. And so what we're seeing basically is that, you know, just like what others have now surmised, including Dave McDermott, Chuck, others in the field, is that in RCC and I would say in certain other GU tumors, but a little less so, the myeloid population reigns supreme. Okay, so it's clearly, you know, the determinants of innate resistance as well as future resistance really does hinge on this population. So what I basically see is what we see in you know, data that we haven't shown, also additional papers, is that this myeloid population goes hand in hand with this tissue resident T cell population. So when you have one, which is sort of like the tumor killing permissive, you know, thing. We don't really use M1 and M2, but more like this tumor anti-tumor type M macrophage phenotype. It's permissive. The environment is permissive for the expansion of tissue resident T cells, but not so much for the other types. So it really is a specific like microenvironment that controls the tissue resident phenotype. And these are in the strongest responders, right? In the mixed responders, what you see is stuff in between. If you actually saw sort of the pseudo diagram, you saw sort of a mix. I didn't have time to go over the mixed colors, but you see partial, you know, differentiation of those types of things. Populations into the NK phenotype, but you see a lot go into the exhausted, right? And what you see with the non-tissue resident type is that they all go exhausted and they never revert back, but only in the right myeloid context. So that's our going hypothesis right now, that it is an interplay. It's a rheostat really between the right myeloid population, as others have, and this tissue resident phenotype. It's not the other stuff. It's specific for RCC and the tissue resident repertoire.
That's really interesting. Dr. Kolenko, we've had several questions come up, so I'm going to maybe pull some of these together. One of the overarching questions we've gotten is, you know, what are your next steps for your finding? Do you anticipate trying to translate this to kind of clinical use? And to go alongside that, did you in your models see any increased toxicity or side effects when you combined the sunitinib and the lorlatinib?
Let me answer like the last question first, because I just answered it. When we used a combination treatment in animal model, we didn't see any side effects such as weight loss or any signs of illness. Of course, I cannot speculate what will happen in humans. We didn't do actually a proper analysis of toxicity, like we didn't examine like maximum tolerated doses, but in the course of experiment, when we used agents at concentrations which are commonly used as single agent concentration, we didn't see that combination added any toxicity to sunitinib treatment in animals. What would be next step? As I mentioned, first of all, we developed a technique which can be applied to any basically drug or any tumor model, any disease model. So we would like, of course, like to see, first of all, to do the same. We found that lonafarnib sensitizes tumor cells to other TKIs like axitinib, sorafenib, but we would like to do the same screening using other TKIs, cabozantinib probably, and like some other targeted therapeutic agents like temsirolimus, and to see if there are any overlapping targets for different, including the same pharyngeal transferase. Plus, we would like to see if pharyngeal transferase inhibitors like lonafarnib and tipifarnib sensitize tumor cells to CDK4/6 inhibitors, because according to some recent publications, these drugs like palbociclib, abemaciclib, and like ribociclib, they can also be sequestered in lysosomes. Therefore, by treating cells with pharyngeal transferase inhibitors, we potentially can improve response, tumor response to CDK4/6 inhibitors as well. And actually other drugs which are used and which also can be sequestered in lysosomes. But I'm particularly interested in trying to find like synthetic lethality targets for tumors which lack VHL or which lack active VHL, I would say, because as you know, like about like 90% of sporadic RCC tumors, they don't have functional VHL. Therefore, we will be able to target specifically VHL if we, of course, initially we need to find gene. Next, we have to find gene product and find if there are any chemical inhibitors and preferentially like clinically relevant inhibitors. But there are a number of databases nowadays which you can search and find it. So basically, this is it.
Very good. Well, I think that pretty much brings us to the end of our question and answer session. And I'm going to start by transitioning us over. We now will be, I'll let Eric introduce our next section.
Sorry, my mic was locked there. Okay, so this concludes the first part of our session 3. We're moving on to our second part. We're going to be joined by 4 new panelists who will give 2 to 3 minute short summaries of their full presentations, which are available online using the link provided in your virtual brochure. We will be limiting the summaries to just 3 minutes, so it's going to be short, but this will allow 10 minutes for questions to be addressed to all panelists at the end prior to taking a break at 3:40 p.m. So without further ado, our first panelist, Dr. Shrezi Rathod, is from City of Hope and will be giving a brief summary on his talk entitled Distinct Cytokines That Predict Response to Immunotherapy and Targeted Therapy in Metastatic Renal Cell Carcinoma.
Hi there, I'm Shrezi Rathod, I'm a second-year fellow at City of Hope, as Dr. Kaufman mentioned. I'll be presenting our abstract entitled Distinct Cytokines Predict Response to Immunotherapy and Targeted Therapy in Metastatic Renal Cell. As we all know, the landscape of systemic therapy for metastatic renal cell has undergone considerable change in recent years, with numerous treatment options and no firm consensus regarding their optimal sequence. Biomarkers to select patients who will benefit from systemic therapies are urgently needed. Several groups have explored cytokine concentrations as potential biomarkers for RCC patients on VEGF TKIs. Tran and colleagues retrospectively noted negative associations between overall survival from frontline pazopanib and pretreatment IL-6, IL-8, VEGF, osteopontin, HGF, TIMP1, and E-selectin. As Dr. Drake referenced earlier, Schalper and colleagues showed that in a recent Nature publication, baseline IL-8 may be a negative prognosticator for ICI's effectiveness across several tumor types, including RCC. To further evaluate the efficacy of cytokines as biomarkers in metastatic RCC, we conducted a prospective trial to determine if various pretreatment and on-treatment cytokine levels in peripheral blood predict clinical outcomes for patients with metastatic RCC receiving either ICI or VEGF TKI therapies. In our study, all participants were planned for treatment with either VEGF TKI or ICI. Enrollment was open to patients across all 8 RCC subtypes, IMDC risk classifications, and lines of therapy. Peripheral blood samples were obtained from each patient prior to the initiation of systemic therapy. A panel of 30 plasma cytokines was quantified for each plasma sample using the human cytokine Vitrogen 30-plex protein assay. And clinical benefit was classified as a complete response, partial response, or stable disease in excess of 6 months. I know it's kind of small on the screen here, but we analyzed 56 patients, 40 of whom were male, 16 of whom were female. Median lines of therapy were 2. Most common ICI was nivolumab monotherapy. Most common VEGF TKI was cabozantinib. 7 patients achieved partial response, 25 achieved stable disease, 17 had progressive disease, and 7 discontinued treatment due to toxicity. Patients with clinical benefit from VEGF TKIs had lower pretreatment median levels of IL-6, IL-1 receptor antagonist, and GCSF compared to patients with no clinical benefit. No significant pretreatment cytokine differences were seen between patients on ICIs. After 1 month on therapy, patients who derived clinical benefit from ICIs had higher relative values of interferon gamma and IL-12. For patients on VEGF TKIs, those with clinical benefit had lower relative values of IL-13 and GM-CSF, as well as higher relative values of VEGF as compared to those without clinical benefit. In conclusion, pretreatment IL-6, IL-1 receptor antagonists, and GCSF, as well as on-treatment IL-12, IL-13, interferon gamma, GM-CSF, and VEGF were significantly associated with clinical benefit from either VEGF TKIs or ICIs. This unique parallel analysis of VEGF TKI and ICI regimens support the further in-depth study of plasma cytokines as biomarkers for immune phenotype stratification in order to provide more individualized treatment and improve patient prognosis in metastatic RCC. I'd like to thank my mentor, Dr. Monty Powell, our biostatistician, Nora Ruel, our chair of medical oncology, Dr. Ravi Salgia, and the rest of our research team at City of Hope. Thank you.
Thank you so much for that talk. It's now my pleasure to introduce Dr. Johanna Bindel at Sarah Cannon, who's going to discuss updates from Checkmate 920, looking at alternative dosing of nivolumab, 6 mg per kg plus IPI, 1 mg per kg every 8 weeks, alternating with nivolumab 480 every 8 weeks.
Thank you so much. It's my pleasure to be here to present to you this data from nivolumab, 6 mg per kg plus IPI, 1 mg per kg every 8 weeks, alternating with nivo every 8 weeks, 480 milligrams every 8 weeks for first-line advanced renal cell carcinoma, Checkmate 920. Here are the disclosures and study background. So we do know from the Checkmate 214 trial that nivo 3 plus IPI 1 Q3 weeks for 4 doses, followed by nivo 3 Q2 weeks, has shown long-term efficacy and tolerability for patients with first-line advanced renal cell carcinoma, and that's the current approved dosing for patients. And so this trial was a community-based multi-cohort phase 3/4B trial that was conducted to look at nivo IPI dosing in predominantly clear cell renal cell cancer patients, as well as standard dosing in patients with clinical features that were mostly excluded from the phase 3 trials. And so here we're going to look at the modified nivo IPI dosing cohort. Here's the study design showing all 4 cohorts, and of course the data here is presented from cohort 1. Looking at the baseline characteristics, we do see that the IDMC risk group percentages are about what we saw with other larger randomized studies, including Checkmate 214. You can also see here that 11.3% of patients had sarcomatoid features. Patient disposition, most of the patients did come off study for eventual disease progression rather than anything that was toxicity related. Looking at dose intensity, we definitely saw that most patients were able to receive most of the dose intensity and were able to receive a large number of doses of the clinical trial drug. We did not see any significant difference in immune-mediated AEs occurring in one patient or more with the advanced renal cell carcinoma with this combination and at this dosage. Here's another look at treatment-related AEs, and again, nothing surprising in terms of what we saw for this treatment. Here are AEs leading to discontinuation, most of which were more of a neoplasm progression or potentially related to very rare autoimmune issues. Looking at response rate, you can see here an investigator-assessed overall response rate that was 34.4%, which is about in line, maybe a little lower than what we've seen in the CheckMate 214 protocol. Here's progression-free survival with a median progression-free survival of 4.8 months, which is a little lower compared to the CheckMate 214 results. And overall survival, median overall survival is not yet reached. So when we look at the summary of the efficacy in CheckMate 920 versus 214, you can see here that in this community-based trial of a different dosing schedule, as well as dose of nivo/ipi for patients with first-line advanced, primarily clear cell renal cell carcinoma, we really didn't see a significant improvement over data that we saw from CheckMate 214. And in summary, because of this, we don't see a reason to change the recommended dosing from the dosing from CheckMate 214. And of course, we have to always be careful to make comparisons between a single-arm community or phase 3B/4 community-based trials as to a randomized phase 3 controlled trial. So for this particular regimen, we would not recommend necessarily using this new dosing and schedule, but continue with the standard combination dosing that was in the pivotal CheckMate 214 trial. And here are my acknowledgments. Thank you so much.
Excellent talk, Dr. Bendel. Next, Dr. Nizar Tanir from MD Anderson Cancer Center will speak on 4-year follow-up for CheckMate 214 with a subgroup analysis of patients without nephrectomy.
Thanks, Eric. It's a pleasure to present the updated results of CheckMate 214 that Joanne alluded to. And also focus on a subgroup analysis of patients who had primary renal tumor in situ at the time of enrollment on CheckMate 214 and the outcome. We all know that patients who have primary in situ and have metastatic disease have a worse outcome, and looking at the effect of nivo/ipi on these patients is of interest. These are my disclosures. So nivo/ipi was approved based on the results of CheckMate 214, and the primary endpoint was PFS, OS, and ORR in patients with intermediate risk, poor risk. We know the standard dose and schedule of nivo/ipi and the sunitinib, and this abstract and this presentation, which was also presented at ESMO this year, looked at, as I mentioned, a subgroup analysis of patients who had primary renal tumor in situ. This was a unplanned, non-pre-specified post-hoc exploratory efficacy analysis of these patients. And here are the two groups with primary kidney lesion: 53 patients with nivo/ipi and 55 patients treated with sunitinib. You see the baseline characteristics, and then side by side the overall population, the intent to treat. Again, nivo/ipi versus sunitinib. The two strata used in CheckMate 214 are IMDC risk as well as region. And here is the summary of the ORR, OS, and kidney tumor reduction in patients with a primary kidney lesion intact. The confirmed ORR for the 53 patients treated with nivo-ipi who had primary renal tumor in situ was 34% and was 14.5% in patients treated with sunitinib. This was a significant difference, although these are small numbers. There were no CRs, as one would imagine, in these two treatment arms. And if you look at ongoing response, about half had ongoing response with nivo-ipi, and then the same with sunitinib. But the median duration of response was longer, was 20.5 months with nivo-ipi versus 14.1. And then when you look at maximum reduction from baseline in target kidney lesions, reductions of 30% or greater in target kidney lesions were achieved in 35% of patients treated with nivo-ipi versus 20% treated with sunitinib. And on this slide here to the right side, you see the Kaplan-Meier curves for OS for these two groups. And as you can see here, the hazard ratio for the difference in OS between these two groups is 0.63, and the confidence interval 0.4 to 1. And the median OS for the nivo-ipi treated patients was 26 months versus 14 months for the group treated with sunitinib. On the slide here are the OS PFS in intent to treat the overall population, the intermediate risk, poor risk group, and the favorable risk. And as you can see here, for the ITT and the intermediate risk, poor risk, nivo-ipi performed very well. The median OS for the ITT was not reached. This is minimum 4 years now of follow-up versus 38.4 months median OS with sunitinib that had a hazard ratio of 0.69. And for the intermediate risk, poor risk, which was the primary patient population for CheckMate 214, the median OS was 48.1 months. So 4 years median OS for the nivo-ipi, and the median OS for sunitinib was 26.6 months. Just remember that this is the median OS for patients treated with sunitinib has not changed at the different time points at the initial analysis of 17 months minimum follow-up, 30 months minimum follow-up, 42 months minimum follow-up, and now the latest most extended follow-up of 48 months. So the sunitinib has remained, the OS median has remained the same, around 26 months. When you look at the favorable risk, the median OS was not reached in both treatment arms, and the hazard ratio is 0.93, but the confidence intervals are wide. For PFS now, I'm not showing you the Kaplan-Meier curves again for the time constraints, but PFS outcomes favor nivo-ipi versus sunitinib in the ITT as well as the intermediate and poor risk patient population. In favorable risk, the hazard ratio for PFS favors sunitinib, but wide confidence intervals. So this is a summary of the ORR, best of overall response and duration of response in all these categories, the ITT, the intermediate poor risk, and the favorable risk patient population. Across the board, the nivo-ipi performed quite well in patients with intermediate risk, poor risk versus sunitinib and the intent to treat. You see the ORR was 42% versus 27% in the intermediate risk, poor risk, was 39% versus 32% in the intent to treat. For favorable risk, which was an exploratory group, the ORR with nivo-ipi was 30% versus 52% with sunitinib. This was a significant difference in favor of sunitinib for ORR. Across the different IMDC risk categories, the CR rate was above 10% with nivo-ipi, and you could see it here on the slide. It was 12% in patients with favorable risk, so double the CR rate with nivo-ipi versus sunitinib in favorable risk, and it was slightly above 10% for intermediate risk poor risk and the ITT. I think it's important to note that there were more patients treated with nivo-ipi who had ongoing responses, and this was across all IMDC risk categories. And the median duration of response was also superior in patients treated with nivo-ipi, again across all IMDC risk categories. And as you can see at the bottom of the slide, the median duration of response was not reached with nivo-ipi across all these risk categories. So the safety summary, I think in one word, we did not see any new signals of safety with further follow-up of minimum 48 months on checkmate 214. And then again, in that subgroup of patients who had primary renal in situ, again, at least the results were either similar or even slightly better than for the all ITT patient population. I think it's important to remember then that for the grade 3-4 patients treated with nivo-ipi with, again, 4 years minimum follow-up, grade 3-4 was 48% versus 64% with sunitinib. And in that subgroup of patients with primary renal tumor in situ, 42% grade 3-4 with nivo-ipi versus 59% with sunitinib. Conclusions, superior OS and ORR were maintained with nivo-ipi versus sunitinib in both the ITT and the intermediate risk poor risk patients with extended follow-up of minimum 4 years. In patients with favorable risk, median PFS was longer and ORR was higher with sunitinib, yet the difference, I would say, is not conclusive in terms of OS in favorable risk. With extended 4-year follow-up, responses with nivo-ipi were deep and durable, regardless of IMD risk-based prognosis. In the exploratory subgroup of patients with a primary renal tumor, nivo-ipi offers a benefit in ORR, duration of response, OS, and kidney tumor reduction versus sunitinib. OS benefit is consistent with that observed in the ITT and in the intermediate poor risk patients, and no new safety signals were identified with longer follow-up in all treated patients, as well as in that subgroup of patients with a primary renal tumor. And these are the acknowledgements. Thank you very much.
Enticed by the fact that he get all these resources and other folks to work with, right? And a place that's growing, where he would certainly benefit. I was pretty honest with him. I said, it's just
great. Thanks so much for that wonderful talk. And I'd like to transition to our next speaker.
Right, which you may not be getting over there. So.
Hopefully you all can hear me. I'd like to introduce a colleague of mine, Dr. Matthew Tucker, here at Vanderbilt University, who's going to be giving the highlights of his poster entitled Association of the Neutrophil to Eosinophil Ratio with Response to Ipi-Nivo in Metastatic Renal Cell Carcinoma.
Hi, as mentioned, my name is Matthew Tucker, one of the Hematology Oncology Fellows at Vanderbilt University Medical Center. On behalf of my co-authors, I'd like to thank the Kidney Cancer Association for allowing me to present here with you today. So as we all know, neutrophilia has been shown to be associated with worse outcomes in patients with metastatic RCC and is an integral component of the IMDC risk criteria. Additionally, there have been retrospective studies in metastatic melanoma and at least one retrospective analysis in metastatic RCC that have shown an association of a higher baseline eosinophil count with improved outcomes to immunotherapy. Based on this knowledge, we decided to investigate the association of the baseline neutrophil to eosinophil ratio with response to combination immunotherapy with ipilimumab plus nivolumab. Our hypothesis was that the median neutrophil to eosinophil ratio would be significantly lower among patients with response to ipilimumab plus nivolumab compared to the median neutrophil to eosinophil ratio among patients with the best response of progressive disease. For this study, we performed a retrospective review of 40 patients, and the baseline characteristics, including the IMDC breakdown of these patients, are available in our poster and in our pre-recorded presentation. The primary endpoint of our study was met with a median baseline neutrophil to eosinophil ratio among patients with a partial response being significantly lower at 22.5 versus 51.8 among those with progressive disease, with a p-value of 0.02. Shown on the left are the violin plots of these two groups showing the different distributions. For secondary endpoints, we divided patients into 2 groups of 20 patients based off the overall median neutrophil to eosinophil ratio. As shown in the Kaplan-Meier curves to the left, you can see that the median progression-free survival was significantly longer at 7.8 months in the less than median neutrophil to eosinophil ratio group, compared to 2.6 months in the greater than median group, with a hazard ratio of 0.39 and a p-value of 0.02. Looking at the bar graph to the right, you can see the best response as depicted between the 2 groups. As you can see, in the less than median neutrophil to eosinophil ratio group, there are numerically more partial responses and stable disease, and numerically fewer patients with progressive disease as their best response. In conclusion, the median baseline neutrophil to eosinophil ratio was significantly lower among patients with partial response compared to those with progressive disease. This effect was largely driven by the baseline eosinophil count, which was significantly higher among patients with partial response versus progressive disease, as detailed in our supplemental material. When split into 2 groups by the median neutrophil to eosinophil ratio, there was a significant improvement in progression-free survival. The neutrophil to eosinophil ratio warrants prospective validation to evaluate its role as a candidate predictive biomarker for response to immunotherapy combinations in metastatic RCC. I'd like to thank all the co-authors who contributed to this work, and to my co-fellows at the Vanderbilt University Medical Center, and to all the patients and families who participate in ongoing studies.
Okay, Dr. Tucker, great job. So we're going to take a quick session for questions. Reminder, you can use that Q&A tab on your screen. Hopefully we'll have about 10 minutes here. And I'm going to start with Dr. Shirazi-Raffel. Kind of a 2-pronged question. So regarding the known biology of these cytokines that were associated with the response benefit, do these findings make sense to you, or were they surprised, including the absence of IL-8? And second part, what are your next steps to bring this closer to clinical use, including any future validation efforts?
Yeah, those are important questions. Thanks. Looking through our results, there were some associations that were more surprising than others. IL-6 was a familiar face, as our group previously found a negative association between pre-treatment IL-6 and PFS in patients on pazopanib, which wasn't all too unexpected given the IL-6 JAK-STAT3 axis, which potentiates tumor proliferation. Also, interferon gamma has a known connection to immune checkpoint inhibitors. And previous groups such as Zari's and colleagues have established inverse correlation between interferon gamma and clinical benefit in folks on second-line nivolumab. On the other hand, there were certainly some cytokine associations that we did not anticipate. You mentioned IL-8, that was definitely on the list. In addition, IL-13, which is traditionally considered to be more associated with cancer progression via the STAT6 pathway. We found the inverse to be true, but there is an alternative 15-lipoxygenase 1 pathway, which has been shown to induce apoptosis and inhibit proliferation of cancer cells. So we think that that's a potential pathway that can be further explored with respect to VEGF TKIs. To your second question, the future of cytokines, I think that just taking these individual cytokines and looking at them independently offers some interesting insights. But where the future really holds is looking at more of a multifactorial cytokine signature. I know certain groups such as Tran and colleagues and even Dr. Tenier and his group have tried to develop some of these for VEGF TKIs. And I think that figuring out which of these cytokines, when looked at collectively in composite, can be utilized is really where we're going to be heading in the future. So.
Great, thank you. Dr. Bendell, question for you. What was the biologic rationale behind this dosing strategy? And based on these results, do you perceive the need for additional alternative dosing trials, or have we found the optimal dosing regimen and can focus our research efforts elsewhere?
Would
We might need to come. I'm sorry?
Sorry. Can you hear me?
Yes, go ahead.
Oh, sorry. Okay. That I'm not sure if we would move forward with this unless it was done in a much larger fashion. But I think with this initial data that we see, I'm not sure that we would pursue that further.
Okay. Dr. Nazar, can you speak to the range of the primary tumor regression magnitude that was observed with Ipi/Nivo, such as were there ever more than 50 or 75% size reductions, and were there any differences based on starting tumor size? And related to that, can you extrapolate from these data at all whether Ipi/Nivo might be useful for converting unresectable large primary tumors to operable ones in the clinically localized setting? Should Ipi/Nivo replace sunitinib's current occasional use in this role?
Okay, I'll look you into that email. And
Dr. Tanier, are you there? Okay, I think we're having technical problems there. Dr. Tucker, are you there?
I am.
Can you answer what is the next step here with your work, and should we start using eosinophils to counsel patients, or is additional validation needed first? And also, second part to that, if validation is needed, is there feasibility for mining eosinophil data from the CDC tests that have already been tamed in previous immunotherapy trials, such as Checkmate and others?
So thank you for those questions. I would say at first, I would not recommend using this eosinophil data clinically at this time. It's hypothesis provoking and certainly does require more validation and future work. I'd like to be able to get to the point where we can look at these types of analyses prospectively. But if available, we've had many randomized phase 3 trials that have had correlative work, really impressive correlative work done. So certainly if eosinophil data is available from any of those, I would certainly support and encourage looking into it further.
Dr. Tucker, while we're still on you, why biologically do you think higher eosinophils may correlate with the improved response to immunotherapy that was observed?
Sure. So I think there's still a lot more we need to learn about the role of eosinophils in the tumor microenvironment. But there was a recently published review actually talking about eosinophils in the microenvironment from what we do know preclinically. And there are several chemokines that are secreted by eosinophils, like CXCL9 and 10 and CCL5. These chemokines can actually increase or induce migration of CD8 positive T lymphocytes into the tumor microenvironment. And so that's potentially one mechanism why increased eosinophilia may be synergistic potentially with immunotherapy.
Okay, great. Is Dr. Tanier back on by any chance? Okay, well, I think we're right at 3:45, 1 minute short. Okay, we're 4 minutes over. So why don't we go for our break, and we're gonna have you come back in 16 minutes at 4 o'clock to start our next session.